Hi everybody. Now we are going to see about laboratory methods for detection of foodborne pathogen, conventional and rapid methods. First we will see about conventional method, that is culture-based method. Introduction. The examination of foods for the presence, types and number of microorganisms or their products is basic to food microbiology. In spite of the importance of this, none of the methods in common use permits the determination of exact number of microorganisms in a food product. Although some methods of analysis are better than others, every method has certain inherent limitations associated with its use. Now we'll see detection of foodborne pathogen, which includes rapid and sensitive methods like culture techniques, which is maybe a very slow one, and immunological techniques, which is a very sensitive method, and molecular techniques, which uses probes to detect specific DNA or RNA, sensitive and specific. Here we are seeing the examples for the culture techniques, immunological technique, and the molecular technique. For culture technique, they are using a cubic or electronic counter to count the colonies in the agar plate, and immunological technique like latex agglutination test, and then the molecular technique they use PCR reactions. They use thermocycler for this method to detect. There are four basic methods employed for total number as follows. First one is standard plate count or aerobic plate count for viable cells or colony forming units, and the second one is the most probable number method, which is a statistical determination of viable cell, and the third method is dye reduction technique to estimate number of viable cells that possess reducing capacities, and the fourth method is direct microscopic counts for both viable and non-viable cells. Now we'll see the first method, conventional standard plate count method. In this method, portion of the food samples are blended or homogenized, then it is serially diluted, and then it is plated onto a suitable agar medium, which is incubated at an appropriate temperature for a given time, and then after which all the visible colonies are counted by using a cubic or electronic counter. The standard plate count is the most widely used method for determining the number of viable cells or colony forming units in a food product. The diluted specimens are plated onto the surface of the replica plates with the aid of bent glass rod. It will be like a hockey stick, and here we'll be taking 0.1 ml inoculum per plate, and it is carefully and evenly distributed over the entire surface of the plate. Next, we'll see the advantages and disadvantages of the standard plate count. First, we'll see advantages. Surface plating helps in determining the number of heat-sensitive psychotropes in a food product because the organisms do not come in contact with melted agar. Strict aerobes are favored by surface plating, but microaerophilic organisms tend to grow slower. The disadvantages of this surface plating or the standard plate count method are it is not effective due to the spreaders because especially when the agar surface is not adequately dried prior to plating, which makes crowding of colonies and enumeration becomes more difficult. The second method is most probable number method. In this method, dilution of food samples are prepared as for the standard plate count method. Three serial aliquots dilutions are then plated into 9 or 15 tubes of appropriate medium for the 3 or 5 tube method respectively. Numbers of organisms in the original sample are determined by use of standard MPN table. The method is statistical in nature, and MPN results are generally higher than standard plate count results. This method was introduced by McCready in 1915. Coliform densities in foods, a 3-tube MPN value of 10 was found to be as high. Advantages of MPN technique: it is relatively simple and specific group of organism can be determined by use of appropriate selective and differential media, and it is used for determining fecal coliform densities. Drawback of this method is large volume of glassware is required, especially for the 5-tube method. Next method is dye reduction method. Two dyes are commonly employed in this procedure to estimate the number of viable organisms in suitable products. One is methylene blue and another one is resazurine. Supernatants of foods are added to standard solutions of either dye for reduction from blue to white for methylene blue and from slate blue to pink or white for resazurine. One of the problems of using dye reduction for some food is the existence of inherent reductive substances. Another way of getting around the reductive compound in fresh meat is to homogenize sample by stomacher rather than by using warring blender. By using stomacher homogenates, raw meat was successfully evaluated by resazurine reduction when stomacher homogenates were added to a solution of resazurine in 10% of skim milk. Advantages: it is simple, rapid, and inexpensive method. Only viable cells and actively reduce the dyes. Disadvantage: not all organisms reduce the dye equally, and they are not applicable to food specimens that contain reductive enzymes unless special steps are employed. Next method is the direct microscopic count method. In this method, they prepare smears of food specimen or cultures onto a microscopic slide, staining with an appropriate dye, and then viewing and counting cells with the aid of microscope with the use of oil immersion objectives. And this direct microscopic count are most widely used in the dairy industry for assessing the microbial quality of raw milk and other dairy products. The specific method employed was originally developed by R.S. Breed, so it's known as breed count. This direct microscopic count method consists of adding 0.01 ml of the sample to a 1 centimeter square area on a microscope slide, and following fixing, defatting of sample and staining, the organisms or clumps of organisms are enumerated. Advantage of this method is it's rapid and simple, and the cell morphology can be assessed easily. And the disadvantage of this method is both viable and non-viable cells are enumerated. Next one is its food particles are not always distinguishable from microorganisms. So this is the main disadvantage. And another method is Howard mold count method. So this is a microscope method. They'll be using a microscopic slide which is developed by B.J. Howard in 1911, primarily for the purpose of monitoring tomato products. This method required the use of special chamber, which is a slide designed to enumerate mold mycelia. Next, we'll see about rapid method. For a method to be effective, it should meet the following requirements. Detection must be specific, and the method should detect the desired specific pathogen, and method must be sensitive to detect small number of pathogens, and it should produce a quantitative analysis to determine the severity, and the method should be multiplex. There are many rapid methods to detect the foodborne pathogen. First, we'll see about biosensor. It is a device that converts a biological response to electrical signals, and it consists of a bioreceptor element and a transducer. The transducer converts the signal to electrical current and passes it to the amplifier. There is one type of biosensor which is bioluminescence biosensor, which measures changes in luminescence emitted by living microorganisms. There are two types of bioluminescence found in microbes. One is ATP bioluminescence, another one is bacterial bioluminescence. First, we'll see about ATP bioluminescence. It is used to measure the effectiveness of cleaning surface and utensils. Take a swab sample and combine it with a mixture of luciferase enzyme. The following reaction take place: luciferin plus ATP plus oxygen. In the presence of luciferase enzyme, it produces light. Next one is bacterial luminescence. The gene responsible for luminescence in bacteria is called Lux gene. The gene is introduced into the host-specific bacteria. Detectors are then used to measure the emitted light, and it is capable of detecting 100 cells per hour. Next method is electrical impedance method. Microbial growth causes change in impedance and can be detected using electrochemical impedance spectroscopy. It monitors large number of samples, relatively short detection time, 6 to 24 hours. And another method is antibody-based assays. Here it is highly specific interaction of antigen antibody used for detection of pathogens. Lattice agglutination test, reverse phase lattice agglutination test, immunodiffusion, ELISA, immunoprecipitation. The last method is DNA-based assays. These includes the methods based on the use of nucleic acids for detection of pathogens. There are three main nucleotide-based assays. One is DNA hybridization, polymerase chain reaction, DNA microarray. First is DNA hybridization. DNA probes, sequence of known segments, are hybridized with the target genomes. If the relevant sequence is there in the sample, the probe binds with the portion and gives fluorescence. Target DNA is denatured at high temperature. Probes are labeled with radioactive labels for detection. RT radiography is then used to detect the probe target sample. Next is polymerase chain reaction. Primers designed against pathogenic genes are used to amplify the sample sequences. If the amplified product is obtained, this means that the pathogenic DNA is there in the sample. This method is more specific and rapid than conventional method. Real-time PCR method, just like PCR, but it monitors the change in the reaction as they occur by continuous collection of fluorescent signals. Fluorescent dyes such as cybergreen are used in RT-PCR. As the dye binds to double-stranded DNA, it undergoes change in shape and increases fluorescence. Real-time PCR is being used for viral quantification, drug efficacy, and pathogen reduction, genotyping. The third method is DNA microarray. The target sequence are bound to a chip which is normally glass slide or nylon membrane called arrays. mRNA is extracted from control and experimental cells and labeled with specific dyes, for example, Cy3 and Cy5. They are then hybridized with the target probes attached to the arrays. This is a rapid and efficient method and can detect thousands of specific sequences. Thank you all for watching this video.